Two chapters, chapter 10 and chapter 11. Chapter 10 deals with vision, and chapter 11 deals with motor control and plasticity. So let's go ahead and get started. If I can, there we go. The whole area that you can see without moving your head or eyes is known as your visual field. While the visual field may be wide, an individual's visual acuity tends to be only in the middle of the field of vision. This is because there's a concentration of cones in the middle of the visual field, an area known as the fovea. It's right in the middle of your visual field. When you look at something, normally you're focusing the fovea on whatever it is you're trying to see, and it's primarily cones. It's primarily the color discerning receptors in your eye. And there you go. We're focusing, and this is out of focus. However, even with the concentration of visual receptors in the center of the visual field, there's a spot where the optic nerve feeds out of the eye and blood vessels feed into the eye. This is known as the optic disc, and this area maintains a blind spot in an individual's visual field. Normally, the brain automatically fills in the blind spot, so you don't have to worry about it. That's why your vision doesn't have a dot running around in the middle of your field. Vision comes to humans in the form of wavelengths of light, or photons. The human visual ability lies between 400 and 700 nanometers from ultraviolet to infrared. Other animals have ranges of vision that encompass wider spectrums of light. Bees can detect beyond ultraviolet light. And that's one of the ways that they can follow the path of an individual. They can see the path, and that's how they go back and forth. So a bee will find a source of pollen, and then it will fly back to the hive, and then it will stay at the hive, and the other bees will go to the source of the pollen by following its ultraviolet light that it puts off. And that's the path that they follow. A photon of light comes into our eye with select properties that we will perceive as color. The hue is the wavelength of the light. Wavelengths in the 400 to 500 range are very dark purples to blue. Wavelengths in the 500 to 600 range are greens to orange with yellow in the middle. Wavelengths in the 600 to 700 range are bright reds darkening to black. Since most color is detected as patterns of light of different wavelengths, brightness refers to the amount of white light from the object. Saturation has to do with the amount of light of the select wavelength. Of all the photons reflecting from an object, we're at 510 nanometers. If all the photons reflecting off the object were 510 nanometers, the object would be highly saturated as green. And there you go. That's what green looks like. The eye acts much like a camera. The eye has a transparent sac called the lens that widens and narrows to focus light on the retina. Also leading to proper focusing is the cornea, or the curved outer cover of the eye, that changes the light beam as it enters the eye. The shape of the lens is controlled by a set of muscles around the lens called the ciliary muscles. When the lens is made to widen or shorten to allow for focusing, it is known as accommodation. At birth, about 15% of males and 18% of females have severe enough eye problems to require correction. And this is why you see so many children in kindergarten or preschool with wearing glasses. By age 20 until presbyopia, and presbyopia is when your eyes get old and they stop accommodating as well as they did before, about 48% of females and 32% of males require correction. Now you have to ask yourself, when does presbyopia occur? It all depends on, it has a lot to do with genetics. For some people it's in their 40s. Other people in their 50s, and usually by the at least by the end of your 50s, you need to wear some kind of corrective lenses. I'm 73, so I'll be 74 next week. I'll be 74 next Tuesday, next Thursday. And I started wearing glasses in about 2000, and in which case I was 51 years old when I started wearing them. But I've known people that were in their late 30s and started wearing reading glasses. I still wear reading glasses, but they're getting stronger and stronger just about every year. Anyway, okay, so it's about 20 years of good eyesight. If you're not one of those individuals that needed correction as a child, my wife has an astigmatism, and she's worn glasses almost all of her life. 48% of females, and actually she just got a new prescription last month. So, yeah, she keeps having to change her prescription. 48% of females and 32% of males require some kind of correction. The interesting thing is that all my brothers and sisters have worn glasses for probably the last 20 or 30 years. My oldest sister wore glasses all of her life. The rest of us had pretty good vision until presbyopia snuck in, or later, I guess, or earlier, I mean. Nearsightedness or myopia is where the eyeball is too long and the clearest focus occurs before the light hits the retina. Farsightedness or hyperopia is where the eyeball is too short and the best focus point is past the retina. And astigmatism, this is what my wife has, is a misshapen cornea. It's elongated. That causes the light to pass incorrectly into the eye and cause the individual to be nearsighted. Her maiden name is Eurydice, so when she started school, she was way at the back of the room and she couldn't see what the teacher was putting on the board. She couldn't see hardly anything. And so they finally took her to the doctor or to the obstetrician and got her some glasses. And she said she put on those glasses and all of a sudden trees had leaves. She just thought of them as green masses before. And all of a sudden she could see the leaves. And she's worn glasses ever since. The iris is a muscular covering over the cornea that gives the eye its color. Non-brown eyes were a mutation that appeared about 6 to 10,000 years ago. So 10,000 years ago, everybody had brown eyes. The mutation occurred, and now there's a fairly large percentage of individuals who have blue eyes. Let's see if we can find it. Percentage of people in the world with blue eyes. You can see how slowly I type. 8 to 10% of the people in the world have blue eyes, but you'll have to understand that Chinese people don't have blue eyes. Asians very rarely have blue eyes unless they have some European mixture. I think pretty much only Caucasians have blue eyes, or people with Caucasian ancestry, some degree of Caucasian ancestry. So it was a mutation, and it was able to, I guess people found people with blue eyes attractive. And anyway, there you go. The pupil is an opening in the iris that allows light to enter the eye. The movement of the eyeball itself is controlled by the extraocular muscles. And of course, this is a gray eye. The photoreceptor cells of most animals are part of the retina on the back of the inside of the eyeball. The retina is very thin, but still contains 5 distinct layers. The first layer is the optic nerve fiber layer. This is working from the inside out, of course. The second layer are the ganglion cells. The third layer are the bipolar cells. The fourth layer is the actual photoreceptor cells themselves, the rods and cones. And the fifth layer is made up of pigmented epithelium. And of course, it's the pigmented epithelium that shines, that makes animals' eyes, and humans, of course, light up at night. Cats. And of course, if you look at a dog's eyes and they're going blind, you can see it in the reflection of their eyes. Their eyes turn green instead of bright, dull green, not like this cat. The rods and cones detect light that reflects off the pigmented epithelium. The rods and cones release neurotransmitters that synapse with the bipolar cells. The bipolar cells synapse with the ganglion cells that form the optic nerve. And that's the difference between rods and cones. Rods actually look like rods, and cones are actually cone-shaped. While most of the communication in the retina runs from outer layer to inner layer in a vertical pattern, amacrine and horizontal cells communicate with cells in a horizontal pattern. Horizontal cells connect receptor cells. Amacrine cells connect the bipolar and the ganglion cells. The rods and cones represent different functional systems. The scotopic system consists of all the rods in each eye, about 100 million cells. Those are the rods. Those are the ones that see light. The scotopic system is very sensitive to weak light but cannot detect color. Light is captured in the scotopic system by the photopigment rhodopsin. The scotopic system is located primarily outside the fovea. The fovea, if you remember, is a concentration of cones. So the rods make up the scotopic system. The photopic system consists of all the cones in the eye, about 4 million. So you've got 100 million rods and you've got 4 million cones. This system detects color and requires relatively strong stimulation and therefore is primarily a day vision system. The photopic system is located primarily in the fovea but is present in other areas of the retina but in a lower concentration. There are 3 forms of photopigments called opsins that detect the 3 color groupings. The photopigment for the scotopic system is rhodopsin, as we said previously. The photopigment for the photopic system is one of 3 configurations of opsins, as we said previously. All the photopigments in the eye are made up of 2 parts, retinol and opsin. Retinol is short for vitamin A aldehyde, or retinaldehyde, shortened to retinol. Light activates the rhodopsin molecule to rapidly lose its retinol, leaving an enzymatic receptor site on the opsin molecule. The enzymatic opsin molecule combines rapidly with G protein transducin. Transducin, in turn, acts on phosphodiesterase, PDE, to convert cyclic guanosine monophosphate, cyclic GMP, to 5-GMP. Cyclic GMP inhibits sodium channels closing and without the conversion. The sodium channels close. Cyclic inhibits. Oh, it inhibits the sodium channel. So it stops it from closing, and with the conversion, the sodium channels close. The loss of sodium ions depolarize the neurons. I've got a cat that's giving me a hard time. I apologize for misreading things. Since we are dealing with millions of receptor cells at the same time, weak light will only hyperpolarize some of the receptors, and bright light will hyperpolarize most of the receptors. Thus, the response will always be graduated as to the amount of light that is detected. This form of response leads to sensitivity. It allows for adaptation. It allows the individual to integrate the stimulus over time. There are two types of bipolar cells that transmit information: off-center bipolar cells and on-center bipolar cells. On-center bipolar cells are activated when light is turned on. Off-center bipolar cells are activated when light is turned off, so that the eye still has visual capabilities. It detects light coming in from the periphery of the visual field. Photoreceptor cells release the neurotransmitter glutamate. Glutamate is decreased in off-center bipolar cells when light is weak or turned off. Glutamate is decreased in on-center bipolar cells when light is turned on in the middle of the visual field. Each cone in the fovea is connected to 4 types of bipolar cells: one on-center midget bipolar cell, one off-center midget bipolar cell, several on-center diffuse bipolar cells, several off-center diffuse bipolar cells. Each midget bipolar cell connects to just one cone in the fovea. Each diffuse bipolar cell connects to several cones. When light is turned on-center bipolar cells depolarize on-center ganglion cells. When light is turned off-center bipolar cells depolarize off-center ganglion cells. Stimulus only affects the part of the retina that is in the visual field. The retina will create a mosaic of stimulation that the brain puts together as a picture. There are various ways that the eye adjusts to different light intensities. The pupil of the eye will shrink with bright light and widen with dim light. Rods and cones have different thresholds, and this is range fractionation. Photoreceptors will adjust their range of reaction depending on the intensity of the light. This will give the individual visual acuity despite the quality of the illumination. Now, why in the world is all of this possible? Why is this necessary? And the answer is, this is how we have survived as a species over the eons. And it's the only way we've been able to survive, because there are a lot of predators out there, a lot of enemies, a lot of predators, large cats, packs of dogs, that we needed to recognize as being around. So that's one of the reasons why our eyesight is so acute. Calcium is a major player in photoreceptor light adaptation. When an individual changes from one extreme light to another, it takes time to adapt to the new intensity of light. This is because the retinol and opsin break down at higher rates in bright light and must regenerate to be available in the dark. Adaptation is much quicker when adjusting from dark to light because the retinol opsin level is not depleted. And this is one of the reasons why it takes you such a long time. So a lot of people never go into the dark. They hate the dark because it takes too long for their eyes to adapt. But you know, I live out in the middle of nowhere where there's no light. So it doesn't bother me to be in the dark. I can see fairly well in the dark. How do you see well in the dark? Well, actually, all you have to do is wait for 4 or 5 minutes, and your eyes will adapt. And actually, they start adapting right away. It just takes a little bit longer for you to be able to see in the dark. Now, you may see the dark is dangerous, but I've been in the dark a lot. That didn't sound good. I've been in the dark a lot, and I have learned to adapt very readily. I have a brother that I was thinking about this the other day. I have pretty good peripheral vision. I can see just about 180 degrees, you know, from my peripheral vision is really good. I have a brother who has tunnel vision, and his eyes are set back in his head a little bit, and so I can see about 180 degrees. He can only see about 100, maybe 120, 130 degrees. But he can see really well at night because his eyes are set so far back in his head. It's really kind of interesting, and he When he was in the military, one of his jobs was to go out at night uh, and act as a sentry, and he was able to see the enemy long before they ever got there. Because of movement, he could see where other people couldn't. Other people want light turned on, he wants light turned off. We used to play at night, hide and seek and other games you play at night like Pee Wee Stick Down and Kick the Can and whatnot. And he'd win every time because he has the ability to move in the dark, in the shadows where you can't see him. He knows. I don't know. He's just good, and of course he survived Vietnam. It was pretty ugly out there, and it wasn't his country. So, but he was still able to read what was behind him and read what was in front of him. Kind of interesting. Color acuity is more accurate in the center of the visual field because the fovea is almost exclusively cones. Moving out from the fovea, the number of cones decreases and the number of rods increases. Light sensitivity is most acute, about 20 degrees off center, where the concentration of rods is greatest. About half the primary visual cortex, the V1, is devoted to the fovea. And you can see the V1 is right in the back, this yellow portion here. And you can see it's relatively extensive, but it goes into the brain. It is the largest visual field. This is one of the reasons that humans are able to concentrate their special field on tasks close at hand, such as reading and surgery, because of our large V1 area. A scotoma is a gap in the perceptual field, usually due to injury of the visual mapping area of the brain. People with scotoma will often compensate for their problem by allowing the part of the brain not affected to dominate the affected area. And this is known as blind sight. This is what they actually see, and this is what their brain is telling them that they see, because what they have done is just filled it in. Now, if you concentrate on the red plus in the middle of that field, and you do that for about 30 seconds, your eyes will get fatigued and it will start looking like this gray area. This gray area will go away, and we'll fill in with this pattern. So you gotta concentrate on the red dot for about 30 seconds, and you can see it. It just starts going away. Now, what your eyes normally will do is they will move. They will naturally move, trying to take in everything in the field. But because you're concentrating on one area, then your eyes don't do that, and that gray spot will fill in with the pattern that's around there. As weird as that is. Each eye is divided into two parts, the nerves of the temporal half of the visual field and nerves of the nasal half of the visual field. Temporal half, of course, is on the outside of your eye, and the nasal half is on the inside of your eye. As the optic nerve leaves each eye, the nerves come together in an area known as the optic chiasm. At the optic chiasm, the nasal optic nerves cross to the opposite hemisphere, and the temporal optic nerves stay on the same side. After the optic chiasm, the nerve pathways are known as the optic tract. Most axons of the optic tract terminate in the lateral geniculate nucleus of the thalamus. From the lateral geniculate nucleus, the axons radiate into the primary visual cortex, the V1, of the occipital lobe. And we saw how extensive that was and how far it extended into the brain. This is the V1 section. The primary visual cortex is sometimes called the striate cortex because of the striations that run through the area. Visual areas of the brain not in the striate cortex are known as extrastriate cortex. Some axons of the optic tract send their axons to the superior colliculus, where the superior colliculus helps the eye to track rapid movements. And we have this, and other animals don't. Other creatures don't. Why do we need this? We need this so that we can see predators coming at us. And for some people who see movement all the time, it doesn't register with them when they see movement in their peripheral vision. It does for me because I have lived in rural areas a lot. And since I lived in rural areas, there's not that much movement taking place. So when I see movement in my peripheral field, then it's easy for me to detect that. If I were a city person where there's movement on a constant basis around me, then potentially I wouldn't see things as readily as I do because my brain would ignore that movement. The lateral geniculate nucleus, is that right? LGN, yeah. The lateral geniculate nucleus is arranged in 6 layers. The outer 4 are the parvocellular layers, and the inner 2 are the magnocellular layers. P cells have small receptive fields, while the M cells have large receptive fields. M cells are sensitive to low contrast and rapid change. About 10% of the retinal ganglion cells are M cells. 80% of the retinal ganglion cells are P cells. P cells require high contrast to be stimulated. P cells are relatively insensitive to rapid temporal change, such as flicker. P cells are sensitive to different wavelengths of light, and they therefore detect color. P cells have sustained responses. P cells have high acuity. I don't know if you've watched a film or a video, and they'll show a computer screen, and sometimes that screen is flickering. Now, the reason it's flickering is because it actually does flicker. What your brain is doing, it is correcting that flicker when you're looking at the screen. But when they film it, of course, it slows down the flicker. And so then you can see it on the film. The primary visual cortex, the V1, is the first and most abundant area in the human brain. And that's it right there, the V1. From the V1, the stimulus is farmed out to other areas of the brain, depending on what part of the eye the stimulus comes from. Areas are designated from V1 to V5. Researchers have discovered that there are 3 different kinds of cones. Cones that are sensitive to short wavelengths only, cones that are sensitive to long wavelengths only, and cones that are sensitive to the middle wavelengths. Of the middle wavelength cones, some are more sensitive to the higher ranges of middle. Some middle range cones are more sensitive to the lower range of middle. There are twice as many long wavelength cones, reds, as middle wavelength cones, greens and yellows. Short wavelength cones are the fewest, blue and purple. And this is one of the reasons why we see reds so readily, is because so many of our cones in our eyes are long wavelength cones that see red. And that's one of the reasons why blues and purples are so difficult to detect. It's because we have so few short wavelength cones. Myopia is a condition where the eyeball is elongated, causing the image to focus in front of the retina, causing a blurred image. There is some evidence that nearsightedness is caused when children look at images too close to their face, and of course, this happened to my niece because her mother put her down with a bottle, and she gave her walleye because one eye was covered up, and she was just using one eye. So the one that was covered up, the muscles became weaker. And this is what myopia looks like. You can't focus. It's like an unfocused picture. This can be prevented. Myopia can be prevented by only allowing reading in adequate light. Avoid small type. Reading without glasses anytime you can do so strengthens your eyes. And that's what myopia, it gets worse and worse. And that's what myopia looks like. This is what myopia looks like. You can't focus your eyes. Glaucoma is a group of eye conditions that damage the optic nerve, the health of which is vital for good vision. This damage is often caused by an abnormally high pressure in your eye. Glaucoma is one of the leading causes of blindness for people over the age of 60. And my brother has glaucoma, and he has to put eye drops in his eye every morning to reduce the amount of eye pressure in his eye. Now, I've had my eye checked many, many times, and I do not have glaucoma, which irritates my brother because he's also hypersensitive to caffeine, and I'm not sensitive to caffeine either, at all. So it seems like he got all the bad genes and I got all the good genes. Good for me. Okay, let's go on to chapter 11. Let's talk about motor control and plasticity. Since I have the good genes and he has the bad ones. Muscles cover the skeletal system and provide movement. They also provide protection for the organs that lie underneath. How muscles attach to the bones is an indication of the type of movement they perform. Muscles only provide force when they contract, when they tighten up, when they get smaller. So when you make a muscle in your right arm, what you're actually doing is you're making the bicep smaller. And since you're making it smaller, it puffs up, it bends, and now we see a mass. But when we are making our biceps big, we're actually making our triceps small. This gets interesting, doesn't it? Some muscles maintain body posture. Some muscles produce movement around a joint. Any movement that occurs in the body is performed by muscle. Muscles are connected to bone by tendons. Around a joint, there are two sets of muscle groups. When one group contracts, the other extends. These muscles are antagonists, just like your bicep and your tricep. You can't make both muscles contract at the same time. Otherwise, your arm will you'll pull some muscle. So if you are making your tricep muscle, that means your bicep is completely relaxed, and if you're making a bicep muscle, it means your tricep is relaxed. They are antagonists. Muscle groups that act together to perform the same act are known as synergists. Yeah, and this is my son has really big biceps, but I have big triceps, which is kind of interesting because this forms a horseshoe on the back of your arm. So his biceps stick out and my triceps stick out. So if I'm trying to really make impress people with my arm, I straighten my arm, and if he's trying to impress people with his arm, he draws it up. It's really kind of interesting. We're like opposites. And of course, two muscles that do the same thing are known as synergists. Muscles are made of thousands of fibers. These muscle fibers are made up of two kinds of filaments. Thick and thin filaments arranged in a regular pattern. Thick filaments are made up of myosin, and the thin filaments are made up of actin. There are two basic types of muscles in the human body. Smooth muscles contract in an automatic fashion without human control, regulated by the autonomic nervous system. Heart muscles and muscles in the digestive tract are good examples of smooth muscles. The other types of muscles are striated muscles, primarily supported by the skeletal system. And this is what a striated muscle looks like. And this is what a smooth muscle looks like. Since different muscles perform different tasks, each requires different speeds, precision, strength, and endurance. Striated muscle fibers come in two forms, fast muscle fibers and slow muscle fibers. Eye movement must be quick to follow the movement of speeding objects, but only for a short period of time. Extraocular muscles are fast muscle fibers. Fast muscle fibers react promptly and strongly. They fatigue very rapidly. They are used when muscle tension must change frequently. Slow muscle fibers have greater resistance to fatigue, are used mainly to maintain your posture. Muscles react to stimulus coming in from motor neurons. The axon will split into many branches and activate many muscle fibers. As the axon is activated, it releases the neurotransmitter acetylcholine. So acetylcholine is important to movement, and acetylcholine is important to to memory. Acetylcholine makes the muscles react by opening sodium channels and calcium channels in the muscle fibers. The ionic change allows the muscles to contract. The point where the muscle's fiber and nerve structure communicates is the neuromuscular junction. Neuromuscular junctions are large and very effective. That's a neuromuscular junction, right there. Each neuromuscular junction releases enough acetylcholine to not only cause one reaction, but several at the same time. One cause of muscle fatigue is due to diminished effectiveness of the neuromuscular junction. Muscles develop coordination when axon terminals migrate to specific target muscles, and acetylcholine receptors migrate to the cluster axon tips, allowing for more efficient muscle reaction. And that's what training does. Training trains your muscles to be more efficient. Muscles react because spinal or cranial nerves send messages to the muscles through efferent neurons. Motor cells of the spinal cord are larger and faster than other motor neurons. Generally, small motor neurons activate slow muscles and are more easily excited. Large motor neurons tend to activate large muscles, which require more stimulation for activation. I hope, well, there we go, good. Oh, let's see a muscle.
The skeletal muscle of animals, including humans, are striated. They exist in bundles of thousands of muscle fibers. A single muscle fiber is actually a muscle cell. As you can see, muscle cells are very elongated and contain several nuclei. Packed inside the muscle cells are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. When a motor neuron stimulates a muscle fiber, overlapping filaments in the sarcomeres slide along one another, and the sarcomeres shorten. The repeated pattern of the sarcomere results from the arrangement of the two major proteins within the myofibril: myosin and actin. The myosin heads are the actual mechanism for muscle contraction. Now let's examine the process in detail. First of all, when a motor neuron stimulates a muscle fiber, calcium ions are released from the sarcoplasmic reticulum. The ions force the tropomyosin to displace and expose myosin binding sites on the actin. An ATP molecule is unhydrolyzed, and the myosin head forms a cross bridge. When the ADP is replaced by a new molecule of ATP, the myosin head detaches from actin and goes back to its original state. And that's pretty much the whole process. And one more thing. Each myosin head attaches to and releases from actin independently, not in unison. Otherwise, the myosin heads would all release at once, and the whole muscle would go to a relaxed state. And that wouldn't be good.
You want to dance, huh? What a guy. Okay, yeah. Whoops. The body must maintain control over the position of its parts. This knowledge of the position and state of its muscles and limbs is called proprioception. This knowledge is maintained by select receptors that report muscle and joint positioning. Muscle spindles lie parallel to muscle fibers. Golgi tendon organs report on the position of the joint and are situated with one end attached to the tendon and one end attached to the muscle. Muscle spindles are located in many areas of our skeletal muscular fiber. Muscle spindles are actually spindle-shaped. It consists of an afferent element and an efferent element. Their primary function is to maintain your posture and to tell you when you have been in a position too long. When a muscle fiber is activated, the muscle spindle reports rate of change. Rate of change refers to change that the fiber has gone through to be able to function. The muscle spindle also reports force, the force required to maintain contraction. Muscle spindles have two types of receptor endings. Primary sensory endings, wrapped around the middle of the fiber, are activated during the initial phase of contraction and then adapt to a lower discharge rate. Secondary sensory endings, found near the smaller ends of the fiber, are not activated at first, but only after prolonged use. Muscle spindles are informed of impending action through two special motor neurons. Gamma motor neurons, or gamma efferents, inform of change of position from the initial movement. Comprises about 30% of the efferent fibers. Alpha motor neurons perform initial movement. That's about 70% of the efferent fibers. While individual muscle movement is monitored by the muscle spindle, muscle contraction is monitored by the Golgi tendon organ. Golgi tendon organs are able to monitor muscle and joint movement because joint movement is controlled by tendons connected to muscles. And that's what a Golgi tendon organ looks like. Golgi tendon organs are somewhat insensitive to passive muscle stretch because they are connected to the elastic component of the tendon and is only activated under heavy loads. The Golgi tendon organs detect overloads that threaten to tear muscles and tendons. Activating the Golgi tendon organs inhibits the motor neurons supplying the muscles that pull on the tendon and thus, by relaxing the tension, prevents mechanical damage.
Muscle building. The key to building muscle is protein synthesis and cellular repair. Skeletal muscle is made up of long fibrous chains containing proteins. Bundles of thousands of fibers make up the muscle itself. When an athlete engages in a heavy workout, this structure is damaged, causing micro tears in the muscle fibers. The body naturally repairs this damage by mending the torn fibers. The result is a muscle that's larger and stronger than before. And with each additional workout, the athlete adds bulk, strength, and speed.
And one of the reasons that steroids work to build muscle mass is because they allow you to your muscles to repair a lot faster. One of the things that you need to remember is that if you are trying to build muscle, one of the foods that you need to eat is protein. You need every time you lift and destroy muscle mass or destroy your muscle fibers, you need to repair them. How do you do that? You repair them with protein. So you need to consume protein in order to build muscle. Okay. Whoops. I'm sorry. Oh, good.
Muscles. We have over 600 of them. They make up between 1/3 and 1/2 of our body weight. And along with connective tissue, they bind us together, hold us up, and help us move. And whether or not bodybuilding is your hobby, muscles need your constant attention because the way you treat them on a daily basis determines whether they will wither or grow. Say you're standing in front of a door, ready to pull it open. Your brain and muscles are perfectly poised to help you achieve this goal. First, your brain sends a signal to motor neurons inside your arm. When they receive this message, they fire, causing muscles to contract and relax, which pull on the bones in your arm and generate the needed movement. The bigger the challenge becomes, the bigger the brain's signal grows, and the more motor units it rallies to help you achieve your task. But what if the door is made of solid iron? At this point, your arm muscles alone won't be able to generate enough tension to pull it open, so your brain appeals to other muscles for help. You plant your feet, tighten your belly, and tense your back, generating enough force to yank it open. Your nervous system has just leveraged the resources you already have, other muscles, to meet the demand. While all this is happening, your muscle fibers undergo another kind of cellular change. As you expose them to stress, they experience microscopic damage, which in this context is a good thing. In response, the injured cells release inflammatory molecules called cytokines that activate the immune system to repair the injury. This is when the muscle building magic happens. The greater the damage to the muscle tissue, the more your body will need to repair itself. The resulting cycle of damage and repair eventually makes muscles bigger and stronger as they adapt to progressively greater demands. Since our bodies have already adapted to most everyday activities, those generally don't produce enough stress to stimulate new muscle growth. So, to build new muscle, a process called hypertrophy, our cells need to be exposed to higher workloads than they are used to. In fact, if you don't continuously expose your muscles to some resistance, they will shrink, a process known as muscular atrophy. In contrast, exposing the muscle to a high degree of tension, especially while the muscle is lengthening, also called an eccentric contraction, generates effective conditions for new growth. However, muscles rely on more than just activity to grow. Without proper nutrition, hormones, and rest, your body would never be able to repair damaged muscle fibers. Protein in our diet preserves muscle mass by providing the building blocks for new tissue in the form of amino acids. Adequate protein intake, along with naturally occurring hormones like insulin-like growth factor and testosterone, help shift the body into a state where tissue is repaired and grown. This vital repair process mainly occurs when we're resting, especially at night while sleeping. Gender and age affect this repair mechanism, which is why young men with more testosterone have a leg up in the muscle building game. Genetic factors also play a role in one's ability to grow muscle. Some people have more robust immune reactions to muscle damage and are better able to repair and replace damaged muscle fibers, increasing their muscle building potential. The body responds to the demands you place on it. If you tear your muscles up, eat right, rest, and repeat, you'll create the conditions to make your muscles as big and strong as possible. It is with muscles as it is with life. Meaningful growth requires challenge and stress.
Okay, one more. I think. Oh, that was that one. It was. What was the other? Oh yeah, let's see. That was the short one. Okay. Wow, what a memory. Okay, there are two reflexes that are maintained by the spinal cord that are automatic and are not mediated by the brain. The flexion reflex is activated when pain is applied to the leg. The muscles will contract, moving the affected part away from the stimulus. And this is a video showing you how to check how people check their reflexes. Whoops, I'm sorry. I've lost my arrow. There we go. I got it back. There we go. No, this is not that one. Okay, that one's later.
The Hulk is an anthropomorphic exoskeleton which mimics the human form. It provides extra support that enables a person to carry more weight than they normally could. Exoskeleton technologies are considered part of the robotics field. If you want to think about them as wearable robots, that's pretty accurate. They sense what the user wants to do, where the user wants to go, and mimics the motion. We like to call these mantonomous systems. The exoskeleton is primarily composed of titanium components, which are both lightweight and provide the strength needed to carry the load. A microprocessor takes in readings from the sensors that are throughout the structure and calculates where the user wants to move, and then commands the actual hydraulic system to actuate the joints to provide the motion. You can walk, you can run, you can bend, you can crawl, you can leap. It does not impede your range of motion whatsoever. This technology could be a real benefit to the military. Just imagine you're a soldier operating at 6,000 feet in the Afghan mountains and being asked to take 120 pounds up in that level of thin air. How exhausted you would be once you got there. An exoskeleton provides the ability to carry that weight the same distance, but to have energy left to execute the mission once you're there. We've also looked at attachments that allow users to carry weight not only on their back, but also on the front. The exoskeleton will fit anybody between 5'4" and 6'2". The current system weighs 53 pounds, but it's pretty much unnoticeable to the user because what the technology does is it even transfers its own weight to the ground, in addition to the weight you're carrying. And we're taking the load off the soldier's back. And we're going to help prevent injuries that occur from carrying heavy loads and repetitive lifting tasks. The best part about working on this technology is that it gives us a glimpse into the art of the possible.
Okay. In the knee-jerk response, the quadricep muscle that is contracted to allow the knee to bend is informed of tendon pressure that makes the muscle react to stretching, thus the stretch reflex. And of course, if you've ever had a physical, then they did the tendon. I'm sorry, they did the knee-jerk response. The primary lateral cortex, the M1, controls all the muscle movements in the body. That's it right there, that blue line. That's it. The hands and lips utilize the biggest portion of the area compared to the percent of the body that they represent. And of course, that's why I like this picture. It shows you just what are the most sensitive parts of our body. And the answer is our hands and our lips. The M1 communicates with the rest of the body through the pyramidal system. The pyramidal system is a triangular shaped bundle of nerves that runs from the brain through the medulla and into the spinal cord. Lesions to this system will result in the inability to move select joints and limbs. Those neurons that run from the M1 to the muscles are not in the pyramidal system, are made to be in the extrapyramidal system. These neurons control the spinal reflexes, and that's what we were looking at before. Now let's see. Oh, I do have an arrow. There we go. This is the one I was thinking we were looking at before. This is going to show you all the flexion. He's going to check all of her tendons to make sure they all react properly.
The lower body reflexes will be performed in the same manner as the muscle strength exams were performed. We're going to test one side and then quickly test the other side and compare side-to-side responses. We've got 5 reflexes that we're going to go over, one being the knee reflex, one the Achilles reflex, and then the adductor reflex, the internal and external hamstrings. The first one will be the knee reflex. I'm going to have the patient close their eyes, strike the tendon. We got a good response. I want you to keep your eyes closed. Strike it again. Got another good response. Now the Achilles reflex. I'm going to just slightly dorsiflex the foot by reaching underneath the foot and letting her rest the foot on my hand. And then I'm going to reach back and I'm going to tap the Achilles tendon. And I got a real nice plantar flexion of the foot. Comparing side to side. I have the patient lay face down. Now we can do the adductor, internal and external hamstring all in this position. If I'm going to test the adductor, I'm going to have the patient medially rotate the leg. I find the adductor tendon along the medial side of the leg. I've located it, and I'm going to strike over the tendon, and I get a slight contraction of the adductor. For the internal and external hamstring, I just have the patient lay flat. I locate the tendon by slightly having the patient bend their knee against resistance, bring the leg up. I'm going to push down. And the tendons pop out quite easily. I've got them, you can relax the leg. I've got the tendon located, the medial hamstring tendon. I'm going to strike the tendon, and I'm watching for contraction and slight flexion of the knee. On the lateral hamstring, I locate the tendon, and I'm going to do the same thing, strike the tendon, and watch for contraction of the hamstring as well as slight flexion of the knee.
There you go. 5 of them. All right. The extrapyramidal system communicates with the spinal cord through 2 tracts, the reticulospinal tract and the rubrospinal tract. The extrapyramidal system not only handles spinal reflexes, but muscles responsible for breathing and muscles that control the head and neck. And that is it for this week. I will talk to you again next week. I think we're tackling 2 chapters next week, 12 and 13. So, I'll see you next